Thank you, Konstantin. Thank you for the invitation to this school. I regret that I couldn't be with you in person, but I'm still glad to have a chance to talk to you. I think this is a neuroscience school, so I would say something more historical in perspective. Also, some personal involvement in some of the work in neuroplasticity and synaptic plasticity. So maybe some fundamental, but I think it would be of interest to students to know the history of some of the findings in this field. So I would begin. Can you see my slide? Is it clear to you?
Yes, thank you. We
okay.
Can see.
So let me begin. Let's see how. Wait a minute. Okay. I'll begin with a historical note about HAP. Now, you may all have heard of HAP. One of the most influential psychologists and neuroscientists actually in the 20th century. Influential in the sense that he produced a hypothesis that are widely quoted and used and guided neurodevelopment, guided the development of the field. Now, this is a book called Organizational Behavior, published in 1949. In this book, he was considering from the psychological point of view how one could learn from perceptual experience. He made a postulation. Also, this may not, all the idea may not be new, but he summarized it well in this book. That he thinks that the reverberating activity may be triggered by perceptual process, and this reverberating activity may be change the synaptic connections among a group of cells, which you call cell assembly. And this is a way of imprinting the memory as well as in producing a long-lasting memory that can be recalled. Now, in this book, he has one a paragraph that was widely quoted. I think some of you must have seen this. It says that when the axon of a cell A is near enough to excite a cell B, cell A excites cell B repetitively, persistently take part in firing the cell B, then some growth process or metabolic changes takes place in one or both cells, so that A's efficiency in exciting B or in firing B is increased. Now, this efficiency apparently is in the synaptic connections, right? So that was the idea. Now, this is purely a hypothesis, how to translate electrical activity into the a memory trace in the circuits. Now, HAP's postulate was then used to argue that the correlated activity between cells, between neurons, can cause synaptic strengthening or stabilization. Now, this correlated activity in the HAP sense is that one cell A, or a group of cell A, consistently fire a group of cell B to produce activity that are correlated. Right? This is a correlation. Now, HAP didn't talk about uncorrelated activity, but in 1973, Gunther Stent, while he was working on the visual system, trying to model visual system synaptic competition and pruning, he argued that if one has another hypothesis related to this, if one say that uncorrelated pre and postsynaptic activity can cause synaptic weakening and elimination, then you have a symmetrical rules. Right? So this is, in fact, Stan was the first one to spell out these two aspects of the HAPS learning rule. So now we all know that the current simple simplified statement of learning rule is that cells that fire together will wire together. Those who did not fire together will not wire together, right? So that's this Hap's postulate. Now this postulates received real physiological function confirmation later in doing synaptic physiology. One can use find a lot of phenomena fits with the Hap postulate. But in general, we now think that most of the cognitive functions depends on electrical activity, depends on plasticity of the nervous system that involves sensory, motor, or cognitive experience triggered activity that induce the changes in the structure or the function of the neurons and the synapse. Most of us are concerned with synaptic plasticity because activity is very easily can modify the synaptic function. But we also note there are also plenty of evidence that a neuronal property could be also modified by activity, right? So, and also persistently, so the memory traces not only stored in the synapse, but also in the neuronal property itself. I'll mention some findings on this aspect. I think it's a very interesting aspect in my talk later. Right, so once this plasticity occurs, then one can see that the learning memory has accomplished, and there's changes in the cognitive function and behavior in the future. So obviously, the learning process is the this induction of structural and functional change, and the change itself is the trace, is the engram, and is the memory of the learning experience. So the first best confirmation of Hap's postulate is the discovery of long-term potentiation and long-term depression. As you all know, I don't need to say much more about this. Repetitive high-frequency stimulation causes persistent increase in the synaptic strength, and low-frequency stimulation, even though you're given the same number of pulses, could cause a reduction, the depression. Now, in the simple Hap's rule, you can say that high-frequency stimulation, the postsynaptic activity can, postsynaptic activation or potentials can accumulate reach the threshold to fire the postsynaptic cell. So the presynaptic cell will fire postsynaptic cell very much inducing the correlated activity, even though presynaptic occurs earlier, slightly earlier. But if a low frequency stimulation, the postsynaptic potential do not accumulate. So you don't fire the postsynaptic cells. So only the presynaptic cells active. Postsynaptic cells are not active, so you have uncorrelated activity. So this is a rough way you can explain by Hap's postulate. But the people once discovered, once this in the 70s and 80s, this phenomenon of LTP and LTD, a great activity, very active work was triggered on two aspects. One is how the LTP and LTD can be induced. What is the mechanism of induce this process? Now, the basic idea is that the presynaptic cells have to release glutamate in the excitatory synapse. Postsynaptic cells has to be activated. So there's a coincident activity between pre and postsynaptic cell, and the high or low level of calcium that accumulate in the postsynaptic cell resulted in LTP or LTD. And that's the general idea. Well, why calcium can serve both roles? A high level of calcium turns out to be able to activate kinase, calcium, the chem kinase tube in particular, which can produce self-autophosphorylation and persistent kinase activity in the postsynaptic cell. That can be achieved by high level of calcium. But low level of calcium can trigger phosphatase, calcium-dependent phosphatase, which are entirely different substrate. in a way opposite to that of what kinases do. Kinases put on phosphate, phosphatase remove the phosphate, right? No, although not on the same cell of proteins. So this is the way how it was generally think to be LTP and LTD was induced. Now there's a lot of other things going on besides calcium and kinase and phosphatase. There are many other downstream effector proteins. I mean, there are hundreds of proteins in postsynaptic cell that are found to be affected by or related to LTP and LTD. So that was a very active field, and that's still ongoing even to this day. And so several decades of work. Now, the one thing that has been of concern is where is the calcium coming from? Now, in the mid 80s, a great discovery about NMDA receptor channel that was normally blocked by magnesium, but when there's a depolarization induced by glutamate, it removed the magnesium and lets calcium in. Now, this is the primary source of calcium for the postsynaptic modification for LTP and LTD. There are also MPA receptors, which are a sub type of MPA receptor are calcium permeable, which can also lead in some calcium. There are also voltage-dependent calcium channel, obviously. When you depolarize membrane, some calcium goes in through the voltage-dependent channel as well. There's also the calcium-induced release of calcium from stores. And this is a slower process, but it occurs in both pre and postsynaptic cell. It's a very important source of calcium for a delay activation of calcium that triggers many of cellular processes. Now, intracellular calcium stores, and there are also calcium pumps that regulates the calcium. That pumps calcium into the intracellular organelle or pump calciums out of the cell, but this regulates the calcium to a very stable state in a resting state. Okay, so this is one aspect, how the LTP and LTD was induced. But there's another aspect. Before we go that, the LTP and LTD, there's a very nice hypothesis we call it, the BCM theory of how calcium level can trigger LTP or LTD. Now there's this curve, this biphasic curve. When you have the low frequency stimulation, the amount of calcium gets into the cell is low. So that triggers LTD. But the calcium level goes up when you have a higher frequency, then you trigger LTP. So this axis, the horizontal axis, both reflect the frequency of calcium, frequency of stimulation, as well as the amount of calcium influx. And this is the calcium-dependent biphasic modulation of the synaptic efficiency. Now we said that the high level calcium, low level calcium is important. There's good evidence now is that the calcium level above 5 micromolar will trigger protein kinases. Now at a low level of 1 micromolar, it triggers protein phosphatase. Now the resting state calcium is lower than 1 micromolar, fractions of a micromolar. So that's the idea that the two protein kinase, protein phosphatase, they could phosphate the same cell of proteins that regulates the postsynaptic structure and also the ion channels in the membrane that allows the increased sensitivity of the postsynaptic cell to calcium or reduce sensitivity. So the protein phosphorylation could also trigger insertion and removal of the transmitter receptor into the postsynaptic membrane. So all this work that's going on for almost a decade, and now this more or less understood how this postsynaptic and presynaptic changes occurs. Now, one of the most active area for about 20 years, since late 80 to almost early this century, is the problem of expression of LTP and LTD. How we know how LTP and LTD is induced, but how does it expressed in terms of the changes in the pre and postsynaptic cell? There's a lot of controversy or competition in terms of the hypothesis of pre-synaptic change or postsynaptic change. After 2 decades of accumulation of evidence, it's now to many people's mind that both pre and postsynaptic cells are changed during LTP and LTD. Now, for example, there's a good evidence that there's an increase of release probability of presynaptic vesicles. There's also evidence that there's increase of number of release sites of the presynaptic neuron. There's an increase of number of vesicles in the number of vesicles available for release in the presynaptic site, and the strongest evidence, very strong evidence that there's an increase in the sensitivity and the number of postsynaptic receptors. Now, this is in the beginning, this was most dominant theory. Postsynaptic theory is the most dominant theory, but many of the evidence for presynaptic changes also begin to appear later. So this clearly, I think both sides are changed, and the evidence are pretty good for both pre and postsynaptic changes. And there's also, let me see, I'll get this thing out of my view. Do you see this block in view? Okay.
Yeah, we see the
There's
pointer.
also very
We see,
important
yeah.
Huh?
Yes, we
Did
see.
you
Yeah.
see?
Yes, yes.
But I want to move this away. I couldn't see how.
What is blocking your view?
The block in my view is this prompts, all these silencing or sharing symbols. You probably don't see it, right? You don't see it.
No, we
You
see
don't see it. It's fine.
we see the
Okay.
clear screen with the pointer.
Okay, that's good.
Everything is okay.
That's
at our
fine.
side.
Right. So that's okay. Okay. So most importantly, I think that comes in about 20 years or 15 years ago is the coordinated pre and postsynaptic morphological changes. Now, there's very good evidence that the synaptic bouton, presynaptic bouton, and postsynaptic spines undergo morphological changes after induction of LTP and LTD. Maybe not immediately, but within minutes or tens of minutes, there's a morphological change. And this is very important because that alters the electrical current flow, induction of postsynaptic currents. That's very important. So let me see, mention some earlier findings from Bonhoeffer's lab in the late 90s. So they did a very striking experiment in slice culture. So they use a slice culture, put it into calcium-free solution. So there's no synaptic transmission. Then they introduce a local flow by flowing a physiological calcium around some synapse, so they can focus on synapse that's been activated by only activated specifically by this presynaptic stimulation, Sheffer collateral of stimulation, while the other synapses are not active. So that by doing this, they can induce LTP by recording from postsynaptic cell that immediately after high frequency stimulation or theta burst stimulation, they see an increase in synaptic potentials. Right. So this is LTP. But then they follow the image of the synaptic spines. They find that some spines appeared after 10 and 20 minutes after the induction of LTP. Right? So there's a very clear morphological changes by inducing new spines in these dendrites. Right? This is a fluorescent labeled dendrite. You can see that appearance of new spines. Now, spine are postsynaptic site, so you assume that presynaptic Buchang is also there. It's not labeled. So the elevated synaptic transmission may be associated with eventually more synapses are formed. Now we also follow up with the LTD a few years later by looking at the this is in fresh hippocampal slices, calcium labeled, so we can stimulate locally and find the spines that are activated by presynaptic stimulation. Then we follow the size and the shape of these presynaptic or the postsynaptic spines, and look at their spine, the fluorescent intensity as well as the size by looking at the spine head, duration of this diameter of the spine head. We found that in fact, low frequency stimulation, which induces LTD, produce you can see that EPSP is reducing in amplitude, and they also accompany with the changes reduction of the spine head, right? And some spines eventually disappear, not only shrink in size, but eventually disappear. And also we also confirm that high frequency stimulation, you actually increase the size. The spine head increase in size, so it produces the opposite effects. You can produce increase in size and then low frequency stimulation later, and you can then reduce the spine head size. So this shows that there's a structural change. The question is, what does it happen really during the long-term memory process in the brain, in vivo? Now this was begin to be possible to do this type of experiment when 2-photon in vivo imaging now was available. You can open the skull and put a window, a cranial window, and then using 2-photon imaging, not only for the cortex. If you actually dissect the cortex, well, you can get down into the hippocampus and also striatum. And in fact, in this set, we look at only the the auditory cortex associated with long-term field conditioning. Right? You can actually look at the same site, the same spines over 2-week period. If I keep back to the same spine to see the long-term changes of the spine induced during the long-term memory, the field memory process. Now you're familiar with auditory field conditioning, coincident or the CS before US. Immediately before US, you get field conditioning as shown by the freezing response to the conditioned stimulus. This is a well-known field conditioning. Now, the circuits for field conditioning is also well worked out by Ledoux and many other people. In this circuit, we know that the conditioned stimulus from the auditory system arrives at the thalamus and then goes to amygdala. Together, they converge with auditory. The auditory signal goes to the amygdala together with somatosensory, the freezing, the stimulus that caused the freezing of the animal, the electric shock, for example, that converge to lateral amygdala, and then through the process of amygdala, you can have an output that eventually, after conditioning, only the conditioned stimulus would trigger the freezing response. Now, all these circuits were welcome, but there's also a circuit known to the field, but not very much addressed, is that the natural amygdala also sends signal to auditory cortex during the field conditioning. In fact, there's a changes, long-term changes of the auditory response after field conditioning. So we were interested in whether we can monitor this auditory cortical change during this long-term memory, rather than looking at the amygdala itself. We want to see how this long-term memory is stored in the auditory cortex, but this because auditory cortex is much more accessible by two-photon imaging because it's on the surface, right? So what we found is, in fact, there's a structural change. Now, this is 1 day before auditory conditioning. We can look at a projection of axon from the lateral amygdala by fluorescent labeling of neurons in the lateral amygdala. Also, this is a Taiwan mouse, which the layer 2-3 neurons are labeled with fluorescence. So we can look at the spine of the pyramidal neuron and the input from lateral amygdala. So you see these pairs of inputs. And what we found is that very frequently, after 3 days after field conditioning, there's a new spine form on the old pairs of synapse, old synaptic bouton and spine pairs. And that's one type of finding we found. Another type of finding is that on the old spines, a new bouton is formed on the old spine, forming, in both cases, forming multi-synapse bouton. And in this case, with most of the changes, in fact, we found the changes in pre and post synaptic cells are mostly associated with new spine formation or new bouton formation, about half-half, 40-60%. So apparently, these new synapse on the old new elements on the old synapse produces the long-term memory. It's associated very well with the conditioning response. And there's also a spine elimination. Bouton also gets dynamically eliminated, but the elimination doesn't change with time after learning. But the addition, the new synapse, the synaptic pairs appears clearly. So this is structural change, right? So it's also in the adult mouse cortex. Now, that's in adult cortex, but in developing cortex, we know that there's a lot of changes in connectivity, not growth changes, not just synaptic local modification, but during the early postnatal years, there's a great increase in connectivity and large number of synapses are formed. Many are undergoing pruning. This is a well-known process during development. Synaptic density or spine density increases with time after birth. And then there's a substantial pruning, but the net amount is increasing until adolescent period. And abnormality in this pruning process will cause pathological conditions such as autism and schizophrenia. And this is clearly important process during development. And also degeneration with time, with age is related to the normal aging as well as serious neurodegeneration related disease. So we can sort of summarize how the plasticity, synaptic plasticity differences between developmental and adult plasticity. In development, there's large-scale rewiring in the developing brain. But so far, the evidence suggests that plasticity in the adult brain can undergo learning and memory, but they involve very limited large-scale circuit rewiring. And I think it's more likely that it's all local addition of a few more spines, more boutons, making the original wiring persisted. The original wiring is still there. And I think this is important because the rewiring really is important for the entire development of intelligence, cognitive process, all developed through this rewiring in the early age. Now, if we do in the adult stage, we want to preserve this early experience without erasing them, but we still want to allow limited plasticity and synapse for learning and memory. So I think this is a very good compromise during evolution. That adult plasticity is limited in structurally, but early large-scale rewiring can occur. But on the other hand, we also know that after injury, there's a large scale rewiring due to the sprouting of uninjured axons that makes new connection to make connection with neurons that have depleted their inputs. All these are also evolutionary useful, even though it's helpful for recovering the circuits after injury, but it is also the precision is not as good. And it doesn't have the early experience to shape it into the proper circuit that are formed during early development. So that this is actually important plasticity aspect. I think recent evidence in the mouse model suggests that this developmental plasticity, even if you don't have an injury, you might be able to reactivate it by changing the synaptic environment. For example, you can change the EI balance or removing perineural net that consolidate the structure of the synapse. You can use some inhibitory factors for myelin. They all show in the mouse model that they can modify the synaptic, making the synapse more like a developmental. I mean, the evidence mostly comes from the visual system, where the visual cortical binocular plasticity can be reactivated, even though normally it only occurs during in the early critical period. But in adult, you can also reactivate it, binocular rivalry, binocular plasticity in the visual cortex by this process of I just mentioned. So I think this leads to the interesting idea that adult brain may be also modifiable by there are hidden or buried capability that's a developmental capability that can be revived in the adult. Maybe that have some implication not only for therapy but also for in terms of cognitive enhancement. So let me go to the next phase of my talk, which I think we'll begin with actually the real talk today about spike time independent plasticity. Now, we were very interested in HAP learning in the late 90s because it explained a lot of phenomenon, cortical plasticity, development of retinal tactile mapping. All this can be modeled by simple HAPS learning rule that cells fire together, can wire together. But now, for example, in the I'm sorry, I think I lost. Okay, I lost one slide. Now, in the retinal tactile system, the retina makes projection into the tectum, into superior collicles, for example. They have a precise mapping so that the rostral and caudal retinal cells, ganglion cells, mapped to the medial temporal cortex, respectively. So this mapping is accomplished by converging retinal ganglion cell axons to the tactile cells, and that converging in the beginning is rather diffuse, and then activity will shape into a more precise fine-grained mapping. Now, the way that people use the HAPS learning rule is follows. When the near neighbor, when we're doing early experience or visual experience, the visual input are usually spatially and temporally correlated. You see an object, you see certain things moving. The nearby ganglion cells are fired in a correlated manner. So their common input to the tactile cell will be strengthened. Now, distant, if two retinal cells are distant from each other, they tend not to be firing at the same time. They are uncorrelated in their firing because of this spatial temporal correlation of our visual scene. Then they tend to be firing uncorrelated, so distant retinal input will be weakened. So close by input are strengthened, distant input are weakened, so the map gets shrunk and more precisely mapped. So the local neurons produce only converging input to the local tactile cells. So you get a fine-grained mapping just by experience. This was the idea of HAP in the late 90s. That they said, well, this is how the map are formed by correlation. So we were wondering how precisely could the pre and post-synaptic firing in order to consolidate the synapse. Right? You say fire together, wire together, but how closely you have to fire together? 1 millisecond, 10 milliseconds, or 100 millisecond difference in firing could be considered correlated. So we decided to test this in a retinal ganglion cell, which I don't have the schematic to show that process. Basically, we have two retinal input that can be that co-innervate a tactile cell. So in the tactile cells, we can record their synaptic potentials, and we can stimulate one site in the retina, and another site, site 1, site 2, both can trigger subthreshold stimulus, depolarization in the tactile cell. So they are co-innervated. So the question is, how do they, how these synapse modify by activity? So we apply repetitive input 1, repetitive stimulation at high frequency for 100 pulse or so, or we can stimulate input 2 repetitively. We found that there's no change in the EPSCs recorded in the tactile cell after this repetitive stimulation. Now, so this is in a way explained by HAPS postulate, because the presynaptic cell produce only subthreshold input. When we stimulate, in fact, this was a 10 Hertz stimulation that did not accumulate. So every time you stimulate, you have a subthreshold depolarization. Next time it's against subthreshold, you do not fire the postsynaptic tactile cell. So there's no firing of postsynaptic cell. So only subthreshold input at the synapse has no effect on the synaptic strength. Now, if you now change the superthreshold stimulation, now you get a higher stimulation current at the retina. So now more than one ganglion cell, more than a few ganglion cells are activated. So their input to the tactile cells is much higher. So you get a firing, a spiking in the tactile cell. So both input 1 and input 2 are firing the postsynaptic cell. Then you look at 10 Hertz stimulation for 100 pulse, you induce LTP. Input 1 become potentiated. You do that on the input 2, you also get potentiation, even though it's slightly smaller. So suprathreshold stimulation of the presynaptic ganglion cell trigger the postsynaptic firing in a correlated manner. Immediately after the stimulation, you can produce potentiation. It's fine. Now that's HAPS postulate, right? Now the curious thing is that when we do the following experiment, we have a first input was subthreshold. We have a second retinal input that's a superthreshold. Now the stimulation interval is still 10 milliseconds after each other, same interval. Same interval as previous experiment, except the second input is bigger, produce spiking. So in this situation, what we found is that the input 1, which did not excite the tactile cell, become potentiated. Input 2 also potentiated. That's what we expect. Repetitive excitation of pre and postsynaptic or potentiation. But input 1, the postsynaptics, the presynaptic cells are not excited, but postsynaptic cell was firing, right? This is the postsynaptic spiking. So it sort of takes advantage that the presynaptic, this subthreshold input will facilitate it because the postsynaptic cell is spiking, right? This subthreshold input occurs about 10 milliseconds before postsynaptic spiking. They become potentiated. Now this doesn't fit with HAPS postulate because that two cells are not firing together. Now if you turn this around, you find the first cell, first input produce spiking. The second input is subthreshold. Now you found the curious thing that the first input obviously you produce the potentiation as expected, but the second input, which is subthreshold, become depressed. They were suppressed by the spiking. Now still the interval is just 10 millisecond difference. This is a spiking before after the input. This is spiking before the input. They're all 10 millisecond difference. But in one case it produces potentiation, in the other case produce inhibition or depression. Now all these processes are AP5 dependent. It's AMDA receptor dependent. Fits with the LTP and the LTD of the synapse. So you can see how this goes. We immediately charted out all the intervals from a few millisecond difference to 10 millisecond, 20 millisecond, 100 millisecond to see this asymmetry of synaptic potentiation or depression depends on the interval. And the result is the spike timing dependent plot. That the input, if it's before about 20 milliseconds before the postsynaptic cell spiking, it's potentiated. If it's after the postsynaptic cell, it's depressed. So both potential and depression are produced by correlated firing about 10-20 millisecond difference. So both input are both correlated. It's just the order of pre and post synaptic firing are different. Right? So this suggests we have to look at the HAPS learning rule from different way. HAPS learning rule is always on the left side. It's always presynaptic before postsynaptic. Presynaptic input that produce depolarization of postsynaptic cell and fire the postsynaptic cell, right? It's always on the left side. But if you reverse the order, it's actually opposite, right? So this led to we immediately confirmed this also in the culture hippocampal neurons, which show that this order of spiking is very important. Not only that, it only occurs on the input of glutaminergic input on glutaminergic neuron, but not on the GABAergic neuron. Right? Postsynaptic cell types matters, and also the amount of synaptic baseline synaptic amplitude. If it's lower, you get a higher potentiation. If it's already high, the potentiation is small, right? So it's a amplitude dependence, not only spike timing dependent, synaptic strength dependent, postsynaptic cell types dependent. Right? So this is what we now call synaptic temporal specific Hap's learning loop. I should mention that this finding of asymmetry begin with Henry Markram and Burr Sackman's study on the slice culture about on the cortical slice recordings that they show this asymmetry of LTP and LTD, and Curtis Bell and Dominic DeBant also show this about the same time this asymmetry. And we actually show this in vivo in our synaptic competition studies. So it basically confirms the fact that correlated pre and postsynaptic activity may either strengthen or weaken the synapse, but it depends on the temporal order of spiking. Pre before post strengthening, post before pre weakening. And there's a critical window, that's a very important. Window is very narrow, and in earlier work by Henry Markram, it was tested. He tested a window, only very long window of 100 milliseconds. It shows no effects, but in fact, the window is as short as 20 milliseconds for LTP. Although LTD window could be longer, right? So this is now called spike time independent plasticity. Now it turns out that this happens all the places. If you look in the central nervous system, this is in the CA1 hippocampal synapses. You see this asymmetric biphasic LTP and LTD. You see they're also in the spine. In the visual cortex, you see this biphasic LTP, LTD, but also the input location of the on the dendrites seems to matter for the LTD. The LTD window is longer for more distal synapse on more distal dendrite than the more proximal dendrite. And also in the cortex, if you check the same neuron, the same pyramidal cell that innervate two different interneurons. So it turns out that we found that this input actually can produce also spike time independent plasticity. So you have two collaterals of the same neuron. These two collaterals are very different. For low threshold spiking neurons, it's facilitating. If a repetitive spiking presynaptic cell, you produce a facilitating postsynaptic response. Fast spiking neuron, you have a rapid depression of the short-term plasticity. Now, so these two terminals are different on these two cells. The LTP and LTD are different. So it turns out that LTP and LTD window can be observed for the parietal cell to fast spiking neuron, but not for the same neuron, no LTP for the other terminal, for the depression, for this facilitating terminal. Right? This is interesting because facilitating terminal suggesting presynaptic property is different. So if you have a different presynaptic property, you can determine whether you produce LTP or LTD. Right? It depends on the release probability, all those things. So this is another evidence for the presynaptic dependence of or the expression for this long-term potentiation depression. And you can also find this asymmetric, find this spike time independent plasticity of a GABA synapse, although this seems to be more symmetric than glutaminergic synapse. The paradigm is the same. You just put an input, the GABAergic input, you have to measure the synaptic currents or synaptic potential. It's a GABAergic synapse. It's very complicated to look at the mechanism because it's, the changes reversal potential, the potential, the inhibitory input could be depolarizing, right? So this is so how do you determine potentiation? It's not as easy as, not as straightforward as excitatory synapse. But in any way, we can look at the currents amplitude through the chloride channels. You can see that currents can actually depend on the order of pre and post synaptic firing. And it turns out that this was the mechanism totally different. Even though calcium influx seems to be required for this plasticity, it actually involves the changes or the expression of this potentiation depression depends on the chloride pumps that changes the intracellular chloride concentration, which in turn determine the reversal potential for chloride currents, determine the strength of the chloride-induced, the GABA-induced depolarization or hyperpolarization. So if we look at all the synapses that has been reported, this is actually summarized about 10 years after this STTP discovery. It varies a lot for the LTD window. You can see LTD sometimes very narrow window, sometimes very long window, but the LTP window is very close. This is interesting. LTP is usually about 10, 20 milliseconds. In rare cases, a bit longer. This happens both in vitro and in vivo in the various cortical systems. So my next talk related to plasticity is the neurotrophin. Now, this is a molecular mechanism that relates to synaptic plasticity. Now, my interest about neurotrophin derived from my reading of a book in 1985. Now, it's 40 years ago, 40 or 30, 40, almost 40 years ago, by Jeff Nickman and Dale Purvis. This is a wonderful book. It's only published in 1985, never revised. If you look at its content today, I say most of the studies, hypotheses remain untested. The ideas that we had 40 years ago didn't really change that much in neurodevelopment. And that's very amazing. So I had a long conversation with Jeff. Jeff said, a lot of people say, well, it's 40 years ago. Nobody's going to read this book. It's not a good textbook. You really have to revise it. And my advice to Jeff was, no, a classic never needs revision. It's a very good testimony to the field to see how much progress or how much lack of progress the field has been since you have written the book. So he was convinced. I think Jeff is not going to revise the book. And in this book, there's a hypothesis which is still standing, still worth testing. So my experience over the years is that if you want a good problem to work on, read a good textbook, read a classic. There are many classics. There are many good books written by Russian neurobiologists in the past. I begin to read those books now. I think there are many hypotheses, many phenomena that would trigger new experiments and new hypotheses and new tests that are good beginning of research project and also good beginning of new career for young people. So I said, go for this old hypothesis, old findings, and find new experiments. You have to use new technology. There are many new technology you can use to test this old hypothesis, which has been there. Some have been accepted by the field. Some others have not been really tested. I say HAP's postulate, HAP learning rule was in a textbook. It was there for half a century. And I actually started by reading the textbook about Hap's hypothesis. Then we say, well, that correlation raises the question, how much correlation you need to have. So that triggered our set of experiment to look at spike timing. So I think reinvestigate old hypothesis is a good starting point. So let's say in this hypothesis, now I read, also trigger a whole series of experiments in my lab, is that in this book, Purvis and Lakeman produced a neurotrophic hypothesis for synaptic competition. Now, there are many developing synapses, they compete with each other and then prune. They get pruning by rewiring, right? In the neuromuscular junction, each muscle are innervated by 7 to 10 axon, motor axon. In the end, they compete with each other. Inactive axon gets eliminated, active axon persists. In the end, it's one axon, one muscle. Right? So this competition, they say, well, it must be occurring in an activity-dependent manner. How do you change the terminal? How do you destabilize a terminal or strengthen, consolidate terminal? Well, trophic factor. Maybe there's some factor release by the postsynaptic cell. Active terminals take up more, so they survive. Inactive terminal do not take up this factor. They get eliminated. They just die. Right? So that's the trophic hypothesis. Now, in the early 90s, we said, well, why don't we try this? Well, because at that time, there's a group of trophic factor discovered. It's called neurotrophin. You all know the NGF, which is discovered in the early 50s. Levi Montalcini, Victor Hamburger, they produced the first growth factors in the brain. They isolated protein that binds to the receptor. The receptor was later identified. But then in the late 80s, early 90s, a set of other NGF family of protein was discovered. The most important one is the brain-derived neurotrophic factors. They're isolated by Hans Turnen, there's laboratories, Eve Bart laboratory, and then there's also the NT3, NT4 discovered. Now, all these binds to receptors. Now, there are two types of receptors. Specific receptor called TRAC receptor, tyrosine kinase receptors. They bind to specific factors. TRAC A, TRAC B, TRAC C binds to different neurotrophins. There's also non-specific factor called NP75 receptors that binds also to all neurotrophins, but non-selectively. Now, the neurotrophin binds to receptor, triggers dimerization, and it's also known that the non-specific TRAC receptor helps the TRAC receptor function. So there's some sort of interaction between these two groups. Now, since the discovery of these factors, a lot of studies have looked at their signaling. Basically, it is a tyrosine kinase, receptor tyrosine kinase. In other words, this is receptors with the tyrosine kinase activity in their cytoplasmic domain. Now, trigger many pathways in the cytoplasm that then induces gene expression of specific genes. So there's a slow signal transduction process that leading to the neuronal survival, nerve growth. That's the primary functions of neurotrophins. But also, more recently, after this tyrosine kinase pathways work out, there's clear evidence of increasing cyclic AMP and elevation of calcium. Now, this is a much faster signal that we show. In our laboratory, we show very rapid, within seconds of the neurotrophin activation, you see changes in cyclic AMP level and the change in calcium level. So they are fast and slow signaling process. They can be involved in neuronal survival, axon growth, cell fate decision, dendritic growth and pruning, expression of specific proteins. There are many downstream effects. Now, the things of our concern is the synaptic function of plasticity. That's That's our focus today. And there are also effects on sensory perception, cognitive, and behavior. Now, what we found is in the early 90s that if you apply these trophic factors, neurotrophins, at a culture neuromuscular junction, it immediately, within minutes, triggers the spontaneous release of a lot of transmitters from presynaptic side. I potentiate the spontaneous release as well as evoke release. Now you can look at spike triggered postsynaptic synaptic potential. You see that the NT3 and BDNF cause potentiation of the synapse, both spontaneously and also spike evoked response. NGF and the other control mediums doesn't have an effect. So it's specific to this later group of neurotrophin. And this finding that a growth factor, that's supposed to have a slow effects on growth and survival, actually has a rapid synaptic action. It's immediately trigger lots of work on the potentiation, synaptic potentiation in hippocampal slices, in vivo, and also people are immediately look at BDNF knockout mice to see that BDNF is required for hippocampal LTP. And if you look at BDNF, the early developmental cortical plasticity, visual cortex plasticity are also affected. So there are a lot of synaptic effects produced by BDNF downstream processes. And here's an example that LTP-dependent induction requires extracellular BDNF. You just put a BDNF antibody, which just chelate, deplete the extracellular BDNF. There's no synaptic potentiation. It's the same as you apply AP5 that block NMDA receptors. And this is for traditional high-frequency TBS theta burst stimulation-induced LTP, or for spike timing-dependent LTP. They all depends on extracellular BDNF, and also it's a very similar to effects of the blocking NMDA receptors. Synaptic targets are widespread. The BDNF and their family of proteins changes presynaptic transmitter release, transmitter receptor expression, ion channels, gene expression, morphology. Nearly every aspect of the synapse are modified by BDNF. You can show that there's so much evidence that is overwhelming. In fact, if you look at these 4 different aspects of synapse: postsynaptic responsiveness, synaptic morphology, presynaptic release, membrane excitability. That's not exactly at the synapse, but it's also related to synaptic function. All these are modified by neurotrophin, because neurotrophin activity can change neurotrophin synthesis. It can cause neurotrophin secretion. It can affect neurotrophin signaling as well, because the activity produces calcium and cyclin MP that also modulate the neurotrophin downstream signaling process. So in fact, neuronal activity can modulate all these 4 aspects. They could also all going through neurotrophins. Now, that eventually causes the modification of synapse, right? So we call it synaptic plasticity. The process activity that change the synaptic efficacy or synaptic function, we call synaptic modification. This is synaptic plasticity, what we usually say, the activity-induced or activity-dependent modification. But there's another aspect affected by neurotrophin. We call the activity-induced changes in the capacity of the LTP or LTD. Now, this is sensitivity for the synapse to be potentiated or to be depressed. This is the plasticity of the plasticity. It's a metaplasticity. is also affected by neurotrophins. So by the BDNF specifically. So this comes to the puzzling and debating issue in all synaptic plasticity studies. When we talk about a molecule that can modify the synapse, this molecule could be a mediator of synaptic plasticity, or it could be a modulator of synaptic modification. So mediator of a synaptic modification, such as LTP, LTD, is directly in the pathway of activity. But a modulator can modulate the mediator without being triggered by the neuronal activity. So you can have a BDNF to serve as a modulator. You can have many other modulators. In fact, there are probably tens or even up to 100 different molecules at the synapse. If you change them, many of them can modulate synaptic efficacy. So they could be modulated, but they do not mediate because their level are not changed by neuronal activity. They just modify the proteins, molecules that are in the pathway of induction of LTP and LTD. So, so the question we can say in the past 20, 30 years, you see so many papers saying that such and such a thing is required for LTP and LTD. Sometimes you even say, oh, this is something, some enzyme, some protein is mediating LTP and LTD. But those claims for that they are mediator usually doesn't have a complete experiment. Usually, I say mediators. You say you have mediator, you can reduce your pharmacology, reduce this mediator level, or you can activate this level. You can also up or down regulate modification. So they say, well, it's necessary and sufficient, and that's how you say it's a mediator. But in fact, if you have a modulator that comes in that can modulate the mediator, can upregulate the mediator, it can also downregulate the mediator that can also upregulate synaptic modification. So simply by most of the experiment of up and down regulation, you cannot distinguish clearly it's a mediator or modulator. In fact, most of the so-called necessity experiments or the sufficiency experiment, the modification that's being changed due to this manipulation are never 100%. Now you can inhibit the mediator, you probably reduce 70, 60, or even 90%, but there's always something left, right? If you activate this mediator, maybe you can activate some percentage. But that's also modulator can do the same thing, right? That's all the problems of confusion in the molecular mechanism for LTP and LTD, for the induction of LTP and LTD. So the one important issue is that if it's a modulator, it should not be modified by activity, but in fact, in neurotrophin, neural activity changes BDNF. It causes increased synthesis and secretion, and that itself also can modify the mediator. It can also directly modify synaptic modification. So BDNF become a really multiple roles in the synapse. Nobody actually has clear consensus. Actually, most people doesn't look at BDNF trying to figure out to say like it's only a mediator or only a modulator. Many evidence are provided. If you group all together, you can say that BDNF is both a mediator and also modulator, modulator, right? So one thing that you must have heard, the transition from yesterday's lectures, the consolidation of the the synaptic efficacy is the transition from early unstable LTP to a late stable position-dependent LTP. This, in fact, can be effective. There's evidence, good evidence that BDNF provide this transition. So they are modified this transition, and this LTP could be induced by other process, other mediator. BDNF secretion could also modify it, but BDNF serving a modulator role can promote this induction and consolidation of LTP. On the other hand, there could be even separated process that BDNF itself, the secretion of BDNF itself could be producing the protein synthesis. It could be directly induced long-term stable or long-term memory without going through the short-term plasticity. Right? So there are many possibilities that remains to be really dissected out. And also there's evidence that BDNF are secreted. Depends on the presynaptic secretion of BDNF. There's evidence that presynaptic NMDA receptors are activated during high-frequency stimulation or theta burst stimulation in particular. This is very physiological. Theta burst stimulation, phasic stimulation, could produce release of BDNF that can be tested or can be monitored by looking at the secretion process of BDNF. And that is required for LTP production. So you have a presynaptic source of BDNF involved. There are many related BDNF and signaling related, neurotrophin signaling related diseases. Right? This is what raises a lot of interest in the field. I've been looking at least the polymorphism of BDNF. The specific polymorphism affects the processing, the synthesis of BDNF, and the transport of BDNF to the surface. That affects the synaptic function in terms cognitive function. So one can think of a lot of cognitive deficits or pathology could be induced by either the neurotrophin secretion, receptor synthesis, and then their levels in the brain. All these red are data that were collected in the human. People have found in the human studies. Finally, I think I may have 10 minutes, 30 minutes before I end. Let me just relax a little bit.
10 minutes, yeah, it's fine. And
10
just
minutes.
to leave few minutes for questions.
Okay. So let me go into the neural circuits. What does it do? Why do we need spike time independent plasticity? Let me play a game. Let's relax a little bit. Let me show a piano music, and then you guess what it is. Who is the composer?
MU-MING, we think that you didn't turn on it yet. We cannot hear. Can you try once more to play it?
Okay. I can hear here, so
I see.
So I think it's the transmission.
It
Anyway,
might be
I just
transmission,
tell
yeah.
you, yeah, I just tell you, I have a piano music synthesized by computer, 2 pieces. The first piece I play, nobody recognizes except it's a Bach, like Bach, Baroque music. The second piece is everybody hear it. Oh, that's Beethoven. And it turns out that these 2 pieces are the same scores. The first piece are played, computer play backwards. The second piece is the forward, the regular duration. It's actually very simple. It's Beethoven's For Elise. Piano, everybody knows. So the demonstration of this is to say that the memory is temporally specific, very temporally specific. The sequence of events, sequence of notes, and the interval between notes are what we remember. So we need a mechanism to hold the sequence in the brain and also the interval. So let's see, play a thought experiment. Right? Let's suppose we have a group of neurons that are well connected, uniformly connected. A group of neurons connect to the next group, a synchronized group of neurons. We cause them to fire in a synchronized manner, one after another. So this group fire first, and then this group, and this group, right? So you do this synchronized firing, cause a synchronized firing chain in the circuits. The result by the spike time independent plasticity is all the downstream connection get strengthened because it's the pre, before, post. The reverse is depressed. So you become an asymmetric circuit. So once you have the asymmetric circuit, you now you trigger only the beginning neurons. They can use this store memory to trigger the subsequent nodes. Subsequent. I just like remembering the sequence of music. Play a few notes, you know what music it is, right? Because you have a sequence memory, right? So this is how the sequence, the brain can do it. Use spike time independent plasticity to store the sequence memory. So here is the example. So we put we have a tadpole with tactile neuron recorded. You have a light shining to the retina, and you record tactile cell currents. 4 different bars are sweeping across the screen. So each bar creates some synaptic current, spiking of the neurons. So you look at it in the beginning. If you test at low frequency, you see all 4 directions produce the same response. Now, if you give a training with only one direction of bars, and then you test with again with these 4 directions, you get trend directions gets potentiated, greatly potentiated and lasting very long. And it's like NMDA receptor dependent potentiation. And it's long-term potentiation for a few hours. So we actually store the sequence stimulation. It turns out that it requires the sequential activation of retinal cells and their sequential input into the tactile cells. The interesting aspect about this memory, if you do these bars, you allow the neuron, you voltage clamp the neurons, tactile neuron. You do the bars, then you can see that the potentiation created in the synaptic, in the retinal tactile neurons. So the bar is actually creating synaptic potentiation. That actually in the voltage clamp neurons is maintained at high level. However, if you turn into current clamp, which allow the cell to fire, allow the tactile cells fire, it immediately washes out. The spontaneous firing of the tactile cell washes out the induction induced by the bars. Now this is interesting. Both some synapse you can detect is depression because there's a complicated potentiation depression goes on. LTP and LTD of these synapses are all induced by the sweeping bars, are all can be washed out by spontaneous activity when you allow the cell to fire by current clamping the cell. But this is what we know about the mass learning versus space learning. If you take a theta burst stimulation for 60 times, you produce LTP. But if you switch to current clamp, allow spontaneous activity to occur, it washes away. But if you do the TBS again, you are doing current clamp, allow current clamp to proceed, no spontaneous activity, you produce stable LTP. If you take these 60 pulses, 60 bursts, you split it into 20, 20, 20, spaced learning. Then spontaneous activity has no effect. You can produce stable LTP, and it cannot be washed away. So spaced learning somehow produce consolidated memory, and the interval between this theta burst stimulation, there's an optimum interval. Like in this particular case, now 5 minutes interval is you can get 100% potentiation. Too short is like a mass stimulation. Too long interval, the effect doesn't accumulate. The same thing's true for bars. If you do bar stimulation, you can do a massive stimulation of the bar. You produce potentiation of the synapse that doesn't last. If you space the bar stimulation by a proper interval, it produces long-term potentiation. So this gives us the idea that we now in the synapse, we actually had a phenomenon of space learning, which requires very precisely the optimal intervals for space learning. And that may be different from different type of stimulus, different types of learning. So I'm closing to the end. I will not show this sequence learning in the mouse visual cortex, and also in the motor cortex, the motor learning. In the striatum, the striatum neurons actually, in order to learn to respond to a cue, to move a bar, the motor, the striatum neuron, the dorsolateral striatum neuron, have to fire in a proper sequence. And this sequence of firing, beginning, there's no coordination, but after a few days of learning, the sequence firing appear in both D1 neurons and D2 receptor neurons. And this sequence firing is specific to the motor learning. So you do need producing this sequential, during the learning process, sequential firing is somehow producing synaptic plasticity and the activation of neurons, so that once they learn, this neuron will fire in a sequential manner to produce in direct pathway and different sequence in the indirect pathway. So finally, let's say neuromodulation. Because most of you probably have heard that there's a very active field now about using non-invasive neuromodulation, such as transcranial direct current stimulation, transcranial magnetic stimulation. They're all stimulation that seems to promote a lot of brain functions. Now, how does it work? They are non-specific activation or inhibition. So we thought we should test whether this non-specific stimulation actually modifies specifically some circuits. So here is the mice learning to do motor learning on rotor rods. They fall off from rotor rods, but with time, in a few days, they keep longer time in the rod, rotating rod. So they learn how to coordinate themselves. So this is a motor learning. And this motor learning turns out can be promoted or enhanced by TDS stimulation if you apply stimulation while the mouse is running. If you apply the stimulation while the mouse is not running, there's no effects. There's no promotion of the learning. In other words, non-specific stimulation promotes specific learning if you activate that circuit specifically by performing the task. Now, this is what we learn, and this is very, very specific. If you learn it, motor learning or running the rotor rod, you get asked to do a different beam walking. It doesn't learn. The sequence or the motor technique it learned on the motor learning with coincident TDCS stimulation cannot be transferred to other type of motor performance. Okay, so if you look at the activity of a neuron, it turns out that neurons, you can look at a firing, TDCS, and nodal TDCS depolarize the neuron. It causes the increased firing. Cathodal TDS inhibit the neurons by changing the membrane potential. This is what we now know about how the white current can do. It just shifts the membrane potential to be more depolarized stage. But why it can promote motor learning? Because task-activated neurons, by performing a task, they can be affected more severe, there are more elevation during the stimulation by the TDCS. The firing pattern gets increased. Those are not involved in the tasks. They are not increased by the depolarization of these transcranial currents did not change those neurons that are not involved in the circuit. Those involved circuit, they already depolarized to some extent and firing already. That's further depolarization really enhanced their firing. So you actually activated the specific circuits. That task-specific training is the way that circuit modification can use SGTP, because you wanted to recover, to modify specific circuit. You ask the animal or the human to perform that task. Then you apply non-specific training, non-specific mechanism that promotes the circuits that's been potentiated. So by the spike timing learning rule. So that I should stop there. That's my end of my talk about the neuromodulation. Thank you for listening, and I'm happy to respond to questions. Maybe there's no time.